If you unspool the DNA in a single human cell, you find 3 billion pairs of chemical letters. For 50 years, geneticists believed that almost all of it was useless. We assumed that only a tiny fraction of that code actually mattered, while the vast, dark expanse surrounding it was nothing but evolutionary garbage. In 2003, an international team completed a 13-year, $3 billion effort to map human genetics. The Human Genome Project had finally documented our biological blueprint, but the results were unexpected. We found that humans have roughly 20,000 protein-coding genes. A microscopic water flea has 31,000. We have almost the exact same gene count as a tomato plant. Those 20,000 genes account for barely 1.5% of our total DNA. Because researchers couldn't decipher the remaining 98.5%, they labeled it junk DNA. Broken fragments, dead viruses, and the accumulated wreckage of 3.5 billion years of evolution. This assumption was taught in medical schools and served as the baseline for thousands of research papers for half a century. By documenting only the biological hardware, the genes that physically build our proteins, science overlooked the layer of instructions that coordinates how that hardware actually runs. Nearly half of your entire genome consists of transposable elements. These are jumping genes, sequences capable of copying themselves and relocating to entirely different positions across your DNA. They were first identified in maize by geneticist Barbara McClintock. Her experiments with Indian corn proved that genetic elements could move, demonstrating that our genome is far more dynamic than a static blueprint. Researchers now understand these sequences as a network of control switches. Some have been found to specifically activate developmental genes that facilitated the rapid evolutionary expansion of the uniquely large human brain. Another massive portion of the junk is actively transcribed into non-coding RNA. Instead of building proteins, these molecules act as biological managers, regulating chromosome structure and dialing gene activity up-down. The scientific community has recently validated this discarded code at the highest levels. The 2024 Nobel Prize in Medicine was awarded for the discovery of microRNA, regulatory molecules found entirely within those non-coding regions. The 98% is a massive control board. It determines precisely when and how the protein-coding genes are put to work. In January 2026, researchers at Northwestern University produced maps of how our genome physically behaves. DNA does not sit in a straight, static line. It constantly bends, folds, and loops into distinct, three-dimensional compartments inside the cell. This physical architecture allows the genome to function. By folding, regulatory switches scattered millions of base pairs away can make direct physical contact with a target gene to switch it on or off. This 3D architecture explains a major breakthrough from December 2025 regarding Alzheimer's disease. Researchers found that the genetic variations driving the disease are located entirely outside the protein-coding genes. These variations exist within non-coding regions. They act as regulatory switches for microglia, the brain's immune cells, which play a central role in the progression of Alzheimer's. Understanding disease risk requires mapping more than just a sequence of letters. We have to understand the exact physical shape and folding of the entire genome. The genome is also responding directly to the environment through epigenetics. Chemical marks attach to the non-coding DNA to alter how genes are expressed without changing the underlying genetic sequence. When humans experience severe stressors, like toxins, trauma, or starvation, the body deploys these epigenetic marks as a rapid survival response, rapidly silencing certain genes and activating others. In 1944, Nazi occupation in the Netherlands caused a severe famine. Pregnant survivors passed altered epigenetic profiles to their children. These adaptations didn't stop there. 80 years later, their grandchildren still carry measurable differences in DNA expression patterns, causing elevated rates of obesity and diabetes. Our inheritance includes a physical record of the environments our ancestors survived. We carry the physiological echoes of their lives alongside the code that builds us. This active adaptation extends deep into our history. In April 2026, an ancient DNA study of 10,000 individuals proved that human biology was actively evolving at a population scale just 4,000 years ago. As early populations adopted wheat agriculture, specific DNA variants linked to the immune system and digestion surged in frequency. The dietary shifts of the Bronze Age are still directly affecting our genetic disease profiles today. Because these epigenetic marks sit on top of the DNA, they are reversible. Modern medicine is now developing therapies to remove the marks, turning silenced genes back on without permanently editing the genetic sequence itself. If researchers could dismiss 98% of our biology as junk for half a century, it suggests a significant gap in our current understanding of the human code. It forces us to reconsider the limits of what we believe is scientifically certain.